Okay, thanks for your patience, guys. I think we have it up and ready. Okay, so yeah, as I was saying, if you could go to this link in your browser, it'll take you to a GitHub page where there's some data available for you to download. And then once you open up that page, you'll see a green button called Code. If you click that green button and then click the Download Zip button underneath that, it should download all of these files to your Downloads folder. Hopefully it's a fairly quick download. Someone in the room could let me know if they have that or are having troubles. So yeah, once you get that downloaded, you can go ahead and unzip it. For Mac users, it should just be as easy as double clicking it. For Windows users, right click on that zip folder and click Extract All. And then it should be unzipped into your Downloads folder. Everybody good so far? Once that's unzipped, you can open up that folder and there should be a file in there called tutorial RNASeq.r. If you double click that and you have RStudio, it'll open up in your RStudio instance. Is everybody to that step yet? Got there? Okay. Once you have it open up in your RStudio, if you could highlight the first two sections. So it'll be lines 2 through, I'll have to take a look. I think it might be 14. Just the first two sections, basically highlight those and then click the run command in your RStudio to start running those commands. And this is just going to basically download all of the necessary packages that you need to be able to run the sleuth analysis later on in the talk. If it gives you a prompt that says update all, some, or none, go ahead and type A and update all and hit enter. And then if it gives you any prompts that need a yes, go ahead and give it a yes and hit enter as well. Okay, and with that, I will start in with the rest of the talk. So yeah, the goals for this section are to, are to basically give you guys a nice background on basic RNA-seq analysis, starting with study design and then moving into the RNA-seq data analysis, which includes data cleanup, quantification, differential testing, and visualization. And then as part of that later part of this data analysis section, I'm going to try and lead an in-class walkthrough with real data. And then we'll probably have plenty of time for additional questions at the end. I'm going to do my best to walk through the basics of RNA-seq, but obviously it's a huge topic and you would need days or weeks to really dive into it and fully go over everything. So since we have only a little over an hour today, I'm going to refer you to this paper so you can seek out more answers. And get your details there. Another caveat for this talk is there's really no right way to do an RNA-seq analysis experiment. It kind of just depends on your data. There are so many pipelines and tools out there to analyze your data, and there's been a number of papers that have been published comparing those different types of tools. So I would suggest you go into the literature and take a look around and see which ones seem to appear correct for whatever data you have available. So today I'm just going to show you one of those ways, which is basically a transcriptome mapping approach. So that would be the middle part of this figure here, the green section. And we'll be using a Sleuth and Kallisto pipeline to do that analysis. But again, I would refer you back to that paper to get more details. So yeah, diving right in. So hopefully most of you have an idea of what RNA-Seq is, but if you need a refresher or need a little bit of introduction, it's basically what it sounds like. You're sequencing RNA molecules. Of course, there's many types of RNA molecules and cells, and so today we are specifically just talking about this little red section here, this mRNA. We're not going to be dealing with any of the small RNAs or long non-coding RNAs or any of that. So if you remember your central dogma from your genetics or molecular biology classes, it's basically you have your DNA that gets transcribed into mRNA, which then gets translated into a protein. So we'll be looking at the middle section of that right now with the mRNA transcripts. So sequencing mRNA uses next-gen sequencing to count up the number of mRNA molecules or transcripts that are created from a single gene to answer a biological question, whether that's the response of the gene to treatment, how it responds over time, or just overall profiles and patterns of gene expression. Before you begin any type of RNA-Seq study, I would hope you keep this in mind. I'm taking this from that paper again. This basically says a crucial prerequisite for a successful RNA-Seq study is that the data generated have the potential to answer the biological questions of interest. And of course that might seem really obvious, like duh, but it's really, really important. There's no quicker way to throw away money than running an RNA experiment without thinking it through first. So just a few things to consider. Do you expect large or small effects? Do you expect to see like a very small fold change or a like 3 or 5 fold change difference in the expression level between different treatments? What type of RNA are you interested in? Whether that's mRNA for the most part, most people are, or small RNAs. And then do you have a good reference assembly already, or do you need to build one? So yeah, a big thing to think about is the power of your experiment. So I would suggest you think beforehand of the effects that you think will happen to your system. So effects that are small and expressed at low level will be much harder to detect than something that's expressed at like a 3-fold or a 5-fold difference in expression. So if you're expecting these small effects, you'll probably need to sequence deeper. That just means you'll have to sequence more reads per sample. And or you might need to include more replicates, and by replicates I mean biological replicates, to get the statistical power that you need. And that's kind of what this figure here is illustrating. Basically, you can see that power estimates are given in the middle here in percentages, and as you increase the number of replicates that you include, you get increased power for your experiment, and the same goes for sequencing depth. The more deep you go, the more power you're going to get, and the more able you will be to pull out statistical differences. I would recommend, if you have one available, to go to your statistician on campus, and they can help you figure this out. Or if you have an example data set from your study system, whether that's an RNA-Seq experiment from another species or whatnot, you can use that data to estimate the power yourself using some free online tools, one of which is Scotty. I included the link here to Scotty if you ever want to explore it on your own. Basically, it's a really cool online program that's free, and it basically just pulls in your example data set and then any parameters that you want to give it, whether that's the effect size that you think you'll see or how much money you're willing to spend on your experiment. And if you run that data through Scotty, you'll get kind of something similar to the image that appears here, where it gives you the number of reads going across the x-axis and the number of replicates going up the y-axis. And anything with a red dot basically is a configuration where you won't have enough power to answer your question. And anything with a green triangle is just too expensive. So anything without any of these little features are okay configurations to use for your experiment. And of course, these are just estimations, but it's kind of a nice starting point to give you an idea of how many replicates and how much sequencing you actually have to do. Some other design considerations, library type and sequencing length. This kind of gets back to whether or not you have a nice genome or transcriptome assembly already available. But if not, you may need to do the assembly yourself. And in that case, I would recommend you go for paired end reads, just because paired end reads will help with repeats. It'll help you cross exon-exon junctions and discover new novel isoforms, and will improve your accuracy of detection of low expressed genes. Of course, it costs a little bit more than single end, but I think it's really worthwhile if you are doing your own assembly. Another thing that might be worthwhile, and of course also costs more, is just using longer reads. Most people do use short-readed Illumina sequence for this, but PacBio and Nanopore are always options as well. Another thing to consider is just the tissue sampling. So first of all, you want to make sure that you are considering what time points you do want to sample. That will actually help you answer your question. And when you do sample, you want to make sure you do it correctly. So the correct way to do it is to take that tissue and immediately put it into liquid nitrogen to freeze it. Because if you don't do that and you say take your sample and then run it up to your lab and put it in the freezer, there could be lots of expression differences that are occurring in that small amount of time that will influence the results of your experiment. You also want to avoid any bias due to time of day. So you want to, if you can, try to sample within a short window, all within the same day. And of course, there's many other design considerations to consider, but these are some of the highlights and the main ones I wanted to point out. Okay, so yeah, once you actually set up your experiment, you extract your RNA, you send it off to the sequencing center, you'll get back your FASTQ files. And that's where the actual data analysis portion of this comes into play. I broke this into 4 main steps. Basically starting with data cleanup. So you would like to get rid of any low quality data from those FASTQs by trimming off low quality ends, trimming adapters, and throwing out any short reads that just won't be very helpful or informative. Once you clean up your data, you can move on to the quantification step, which is basically just counting up the number of reads for each gene or transcript after aligning it, of course. And then once you get those read counts, you can move into differential testing. So in differential testing, you're basically comparing the expression between 2 or more conditions for your experiment and seeing if there's a significant difference in expression. And then once you get that, you can actually move into visualization, where you can graph and explore the results and visually see if you can find out any differences. So I am going to walk through the quantification and differential testing and visualization part of this in real time, but data cleanup would just take too long. These trimming programs take a really long time to run. So I'm just going to explain that in a little bit of detail here first. To do data cleanup, I would suggest you run your FASTQs through a program called FastQC beforehand, so you can kind of see what needs cleaning. This program's pretty handy. You basically upload one FASTQ at a time and take a look at it visually. And you can see they have these little tabs on the left here, and they try to mark whether or not they think your data is struggling in those areas or not. So for example, one of these tabs is per base sequencing quality. And I try to give an example of a bad per base quality graph and a good per base quality graph. Quality score is indicated on the y-axis here, and the position in the read is indicated on the x-axis. And it's normal in Illumina sequencers for the quality to taper off over the length of the read, but obviously in this first figure here, it tapered off quite a bit. Like, it's really falling down into this lower quality score region versus a FASTQ file that is high quality and is all staying up within the good quality scores. Another thing to really focus on is the adapter content. So that's this last tab here in this FastQC output. Basically, it will show you whether or not it can detect any of the known adapters. And so you can see in this FastQ here, it's able to detect Illumina universal adapters towards the end of these reads. And so you would know when you're doing your trimming, like, okay, I need to trim off these universal adapters. And so yeah, when you actually do your trimming, there's of course lots of ways to do trimming. Some of the more common programs are Trim-O-Matic and Cut-Adapt. And I would recommend running your data through FastQC before and after trimming, just to make sure that it really cleaned it up the way you wanted it to. So for example, here you can see that the trimming beforehand had a lot of reads that were falling into the low quality scores towards the end, but after trimming it got rid of a lot of those regions. And so you know you're working with high quality data. Okay, so yeah, once you do your cleanup, you can actually get into the quantification step. And so here's where I will attempt to walk you through real data analysis. Can you guys see this? Fantastic. Okay. So this is what we had hoped to have you guys do with us, but it turns out we can't do it. So I'll just demonstrate and you guys can follow along. This is an EC2 instance and it already has all the data loaded onto it. So if I hit this ls command to list out everything that's inside of here, you can see that there's 4 different folders. There's a miniconda, an output folder, a scripts folder, and a studies folder. And by using the cd command, which just means change directory, I'm going to show you all what is inside of the studies folder first. There's a subfolder called module. And if you look within that module folder, you can see that all the FASTQs are in there. And these are already trimmed FASTQs. What's also in here is the reference transcriptome. So today we're looking at just Arabidopsis. This is the Arabidopsis reference transcriptome, the cDNA files. There's no way to get rid of this. So yeah, just moving back up, maybe. How do I clear? So we go back to the top. And then moving into the scripts folder. I already have a script in here called Kallisto.sh, which if you look inside of that script, contains these commands. I don't want you guys to worry too much about the rest of the commands in here, but I do want you to focus on this one underneath index arath transcriptome. Basically, this is the first part of the Kallisto analysis. It uses this index command, and it creates an index. You give it a name from your transcriptome. And then the second part that I want you to focus on is down here. So for every sample in that folder, it's going to run this Kallisto Quant command. And I'll go more into the details of what's actually in that command here in just a bit. So I will start running that. And hopefully it goes. Okay. So you can see there's some output that's starting to appear here. This is the first part. It's actually running that index command here, and this is the standard out that you expect to see if it's working as expected. So I'm going to move back into the slides here and explain a few things while this runs, because it will take a little while to run. Okay. So this is what we've done so far. We started to run that command, and as expected, the output appears. Just to explain a little better what we mean when we say it's indexing. Basically, indexing a genome or transcriptome is similar to like creating an index that you would see in a book. Like if you're opening up a book and you're trying to figure out where in that book like a certain phrase is or where a chapter begins, obviously that would be a lot easier if you had an index to reference, right? Versus just flipping through and trying to find it. And so it's kind of the same thing. It's creating this index that the Kallisto program can go back to and find things more easily. So yeah, once that index command completes, it'll move right on to the Kallisto Quant command. And this is the expected output standout for that. This will run for every sample. So for this particular demonstration, I have 18 samples total. So we should see this quant section appear 18 times. And again, this is kind of what you would hope to see, that it would, run a certain number of seconds and then complete. So like I said, I'm going to explain that Kallisto Quant command a little more. Basically, it's using that index that was created, and we gave it that name AT_transcriptome. and it's pseudo-aligning to that index transcriptome. I also gave it the parameter of having 16 threads to run on so that it would run pretty fast. And then for every sample, I told it to output into this analysis folder with a subfolder that has the sample name. So we're going to see 18 folders show up eventually with the outputs of all of the Kallisto Quant commands showing up in that subfolder. And then at the end, you give it the forward and the reverse FASTQs, basically. And as part of this Quant command, it's going to do these 4 steps. It'll pseudo-align, it'll do the quantification, it'll do a bias correction, and then some bootstrapping. And just to explain pseudo-alignment a little better, I know that's not the most familiar concept. In traditional alignment, the way you do it is you have your reference, and then you have all your reads, and you're trying to match every single base of that read to the reference, right? And so obviously that's a lot of comparisons. It takes a lot of time. That's why alignment is a very long process. But in pseudo-alignment, it just basically asks the question, could this read have come from this transcript? And the way it does that is it constructs a De Bruijn graph where the nodes of the graph are actually k-mers rather than just single bases, and each transcript follows a path along those k-mers. So trying to kind of illustrate it here, this is from the paper where they first demonstrated this. We have this black line at the top, which is the read, and then these colored lines are the transcripts, and then each of these circles is a k-mer. And so you can see each transcript follows a path along this De Bruijn graph where it is intersecting with certain k-mers. And some transcripts share all of the k-mers and some diverge in some sections. And that's what basically creates this k-compatibility classes. And just real quick, just to remind you guys, like, what is a k-mer? The term k-mer is basically referring to a substring of a sequence. And you could have any length k-mer that you want. I'm showing a few examples here of just 7 mers and 21 mers. But yeah, just so you kind of remember what k-mers are. The higher the k-mer, the more unique each subsequence is. And so increased uniqueness equals less chance at connecting the wrong bits together, like if you're doing assembly. But higher k-mers require more memory because you have to store that and run the risk of maybe not overlapping the other k-mers just because they're longer. So it's kind of a, takes some testing sometimes to figure out what k-mer length is correct to use. So yeah, the pseudo-alignment basically uses all this information and is able to run really quickly because it can skip certain bits of information. So that's what these dashed lines are trying to demonstrate here. Basically, if there's these k-compatibility classes all in a row, it's able to skip that information that's redundant and then just, do the alignment much faster than a regular aligner. Okay, I'm not sure if it's quite done yet, but what we should see is a subfolder for every single sample in the outputs folder. Yeah, it looks like it's still running, so I'll let it keep running. But just to show you what it should look like, if you went into the outputs folder after it was all done, you would see these subfolders for each one of them. Within each of these subfolders, there'll be 3 files for the Kallisto command. There's this abundance.hf file, which is an HDF5 binary file that contains the run information, all the abundance estimates, the bootstrap estimates, transcript length, and it's in this H5 format because that's what Sleuth is actually going to read in. There's also abundances.tsv file, which is a plain text file of the abundance estimates. It's basically just there so that you can open it up and view it yourself and get that information. And then there's also this run info JSON, which, as it says, basically contains information about the run. So yeah, now that the quantification is done, you can actually move into the differential testing. And here's where I'm hoping everyone can follow along with that. So if you can open up your RStudio instance, and hopefully everything's done installing there. If anyone is having troubles, could you please raise your hand and Alex can come around and help troubleshoot. But yeah, hopefully it installed Sleuth correctly for you and you're able to run it. If it did, this is what you should more or less be seeing. So we'll just kind of move through each one of these. I'm going to run the sample ID command and then take a look at the variable that it created here. As you can see, it created this variable that contains all of the names of each one of these samples. So there's 18 samples here. There should be 18 names. And it stores it in a variable. Another thing that Sleuth is going to need is the file paths to each of those result files. So that's what this caldirs command does. It goes through that analysis folder and pulls out the paths, and that's what's stored in that variable. And then it also needs to pull in information about the experiment itself. Just to give you an idea of what that looks like. Oops. This Kallisto demo.tsv file contains information about each of the samples that we're looking at in terms of its genotype for each sample and then the treatment that was applied to each sample. And so our program is reading in that table. And if we take a look at it, you can see it pulled all that information in for to later be associated with each sample. And so yeah, we're going to pull all this information together with this dplyr mutate command, and we'll take a look and see what we got. Should hopefully list out where the sequences are or where the files are, and then what metadata is associated with each one of those. Is everyone caught up to there, more or less? If you're able to follow along, if not, okay. This next part is basically using PlantSmart. It's a database. It's using the Biomart to pull in information about the Arabidopsis genome. And so if you list out the attributes that it just pulled in, there's a ton of attributes, but if you go up to the top, you can see that it actually pulled in information about the transcript IDs, the gene IDs, and all of the annotation data, which is what we'll be needing. So in this next part here, we'll actually pull out the transcripts, or so the attributes that we're interested in, which includes the ensemble transcript ID, the ensemble gene ID, the description, and the external gene name. And so it pulls that into this T to G variable. And then a quick renaming step. Basically, Sleuth expects your variables to be named certain things. It expects a target ID, an ENS gene, ensemble gene, and so forth. So a lot of this is just preparing the data for Sleuth, including this step, which is basically refactoring it and releveling it, which means you're pulling everything that's assigned as wild type to the top so that it can compare everything to wild type. And then finally, the actual creation of the Sleuth object itself with this sleuth prep command. So you can see it's pulling in the target information, it's running some models. If you did any bootstrapping, it's pulling that information in, and it's creating this object called SO, which is just a sleuth object that you can use for this later parts of the analysis. And this takes a little bit of time, but it should be fairly fast. Or not. Is anybody able to follow along out there with this data, or have I already lost everybody? No, nothing? Okay. While that's running, I can go ahead and run some of these commands to set up the visualization steps. Or I guess I need to wait for it to go before I can run it. This first one is basically, it's pulling in this cow plot part to be able to view the plots in a more user-friendly way. And then once that's done, I will be able to plot it out, and we'll be taking a look at the principal component analysis, these graphs. And just as a reminder of what principal component analyses are, basically this will be reading in all of the transcript level information for each of your samples. And it tries to reduce all that information down into something, into a reduced format so that you can see which transcripts have the most effect on the patterns within the data. And so what we'll end up with is basically a graph with principal components on the X and the Y axis. And we'll do that for both the genotype variation and for the treatment. And the reason you do this to actually view the PCA plots is to see whether or not your data is behaving as expected. If you have any where you expect them to be grouped together in that PCA plot and they're not, then maybe you realize that you have an outlier in your data, something weird happened, and you should either take a closer look at that sample or throw it away. Okay, this is taking much longer than expected. Yes, sorry. Oh, one second, I'm sorry. I'm going to give you a microphone.
Are you using those PCA clusters instead of like a correlation matrix then?
Yeah, I would use both.
Okay.
It's just a nicer way to quickly visualize the data in a little bit different format. Sometimes correlation matrices are a little bit hard to parse.
Yeah.
It's pretty similar though. Yeah, it's a way to detect outliers.
That makes sense. Yeah, because I didn't use PCA when I did mine, and now I'm like, but I did these like correlation things, and things are grouping the way I wanted them to.
I mean, in that case, yeah, you were fine then.
Okay.
Yeah, you didn't do anything wrong. Okay, I finished. So yeah, like I said, we'll actually plot it out and take a look at these PCAs. Gosh, this window is bothering me. Like I said, so these are all our 18 samples. They're split up by genotype, so you have the wild types in pink, and then the two mutant lines that we have here in green and blue. It doesn't seem to be splitting. I mean, maybe there's a little bit of grouping where the ROS1/4 mutants are split from the wild types. There's not a lot of huge amount of grouping here, but when you take a look at the treatment groupings, you can see, as expected, the ones that received a mock treatment and the ones that received an ABA treatment are really separating out as would be expected. We can also take a look and see which transcripts are contributing the most to those PCAs. So you can get an idea of like if there's any weird outlier transcripts in your data too. And then you can also take a look at those. So like this one here, this is obviously a transcript that's weighing heavily into this principal component 1. So if we take the ID of that and then plot it out, it'll actually show us the expression of that transcript across all of the different genotypes. So here's the wild type, the ROS1/3, and the ROS1/4s. Obviously, like I said, there wasn't a lot of difference between genotype, but if you look by treatment, sure enough, there's a pretty big difference. It's clear that these are much higher expressed in the ABA treated versus the mock treated. So just some ways to explore the data whenever you get around to doing it on your own. This section I will run all together and then flip back real quick to my presentation so I can explain what's going on here. This is the actual statistical part of this analysis where it's actually doing the differential expression portion of it. Oops. So yeah, the first part of this command, it's a sleuth fit command, and it's basically fitting the full model. And this full model, as you can see, it includes this genotype variation S parameter, which is telling it to assume that the transcript is differentially expressed between the conditions, and so it's fitting that model to all the transcripts. You then run this sleuth fit command without that parameter included, and that's basically your reduced model. This assumes no differences between your two conditions. And then once you get those two models run, you're able to run a likelihood ratio test on those two. So you're pulling in your full model and your reduced model into the sleuth LRT command. And that basically goes through, and for every single transcript, it decides which of the two models best fits your data. Is it seem like they are actually differentially expressed, or there's no difference between them? Once that finishes, then we'll have all of the results stored in this sleuth object, and we can pull them out by running the sleuth results command. And we'll pull this into a variable called full results, which we then can take a look at using head. So I'll do that in here in just a sec. And then one last thing I wanted to point out before I go back to RStudio. The likelihood ratio test does give you a p-value, but it doesn't give you any idea of the effect size. So there's no like full difference information included in that, which is why you also need to run a walled test on your data. This walled test, as part of sleuth, will return a b value. This b value is analogous to, but not quite equivalent to, the fold change that you would expect to see. So if we go back, looks like it's all done. So like I said, we'll run the sleuth results command, and then we'll take a look at the data. And so yeah, basically it's a big table of the most significant differentially expressed genes. So here's a methylation gene that gave a pretty low p-value. But as you can see, there's no fold change statistic anywhere in this LRT. So like I said, we want to run this walled test on the data. And then to visualize that, I'm just going to open up this Shiny app. This is one of the main reasons I really love Sleuth, is this easy to use Shiny app where you can just graphically go through and take a look at everything. So I would suggest starting out with the volcano plot. If you're not familiar with volcano plots, it has this B or beta value, which is equivalent to fold change across the x-axis here. And so what you're interpreting here is this is no fold change, and as you go further to the right, you're getting higher positive fold change, and as you go to the left, you're getting higher negative fold change. And then along the y-axis here is basically your significance. So the higher up you go, the more significantly difference there is between the transcripts. And what's nice about Sleuth is, like, for instance, up here, there's this transcript that is very significant and pretty differentially expressed in terms of the fold change. And sure enough, here's that DRM2 gene that we saw earlier showing up in this table down below that you select. And you can select any number, and it'll all show up in this table with the p-value, and then B is your fold change. Another thing to look at within this Shiny, the test table itself, it just basically is kind of similar to what we saw earlier, but this is for the walled test information. So again, it has that fold change value associated with it, and these are all the most significantly different genes ranked here. You can look at, well, if you had a number of transcripts to look at, you could do a heat map. You can also view PCAs from within the Shiny app, which is nice. This is kind of similar to what we saw before, but you can view it by genotype, or if you drop down here, you can view it by treatment. And yeah, you can even do a sample heat map, kind of like what you were talking about. So another way to just view and see if there's any outliers in your data that way. Okay, how are we doing on time? And then finally, if you're interested in a particular gene, you can also do this plot Bootstrap, and it should show up. Maybe not. Oh, it's because I have the Shiny app still open. Don't I? Oh, no. Okay, I don't know why that's not working, but Plot Bootstrap will basically let you graph out any gene, and it should show up within your RStudio here. Okay, are there any questions from that part? Nope.
So my question is, how did your data was normalized?
Was analyzed?
Normalized.
Oh, normalized,
Yes.
yeah. Yeah, there's a couple different ways to do normalization. I would suggest you use the TPM, which is transcripts per million. Is that what you're referring to?
Yeah.
For normalization? Yeah. So yeah, that's a good point. When you're doing these comparisons like this, obviously each gene has a different length, and that can really influence your results. So if you take into account the gene length using normalization techniques like TPM, or there's also FPKM or RPKM, you can get rid of some of those biases, basically. And I would recommend TPM.
Well, in your opinion, that is one that is the best, or in your case?
It kind of switches it. I have to remember how this works. I think it normalizes each transcript to the gene length first, and then normalizes everything as a whole, and it just gives you a little bit better statistical analysis, basically. Yeah. Yeah. Any more questions?
This is kind of a dreamer, naive question, but I'll ask it anyway. So if I understand correctly, if you really want to understand what's happening at different life stages or whatever, really the technology now allows us to capture individual snapshots periodically in time. Do you ever see in the future where we'll have real-time monitoring of RNA in a plant? So you've got it in a special cabinet and it's growing and it's doing its thing. Can you even foresee something like that in the future, or is that just a pipe dream? Actual real-time, over hours and days, monitoring RNA changes?
If by real time you mean like instantly, I don't know of any current methods that would allow you to do that, unfortunately.
But I'm saying where you wouldn't sacrifice the plant, but somehow you would. I'm just wild dream.
I mean, anything's possible, Phil. Yeah. Yeah, I mean, you really don't have to totally sacrifice a plant to even do these studies, right? You just take a section of tissue, more or less. So I don't know if that's as much of the issue. I mean, yeah, you're always going to have to do some level of sequencing to get to these results, at least through an RNA-Seq method. So there's always going to be a delay in that, no matter what. Unless somebody has some really amazing discovery sometime in the future. Is there a way to view questions that come in online? Oh, is there? Oh, there. Oh. Okay.
Okay, so yeah, I have a question. So with advancement in sequencing, we're beginning to get this high quality, sequencing. Do you think it's still necessary at some point when you use fast QC or multi QC to check the quality of your reads and it's perfect? Do you think it's still necessary to do, trimming or anything like that?
I don't know if it's ever going to be a point where you don't have to do any trimming. Even in terms of like adapter sequences, usually they try to trim that off, but you always get the odd instance where you get sequenced through the other end of the read, and so you start to get the adapter on the other side. And as far as I know, there's not great methods to get rid of that from your data set. So at least for that, you would have to term adapter sequence. I suppose if we had a technology that didn't have this trailing effect and just every read was really, really high quality and didn't need that type of trimming, then at least you wouldn't have to trim for quality. But yeah, I think some level of trimming, at least in the current state, is necessary at this point. Yeah.
Darci, I wanted to ask question. What do you think about designing experiments when we're thinking about herbicides and like before and after treatment and what time points? So I know you've worked with these kind of data sets a lot. What kind of things should people think about as far as that part of their sampling design?
Right. No, that's a good question. And I probably should have covered that here. Yeah, so I am a little bit opinionated on this. I think that when you spray a plant and then sample, the differences that you see that are going to come up between a plant that survives that spraying and a plant that doesn't, a lot of those differences are just going to be down to like death processes, right? Like one plant's experiencing the processes of dying and the other plant is surviving. And so any transcript differences that you pull out of those, it's hard to separate that out from like real resistance differences, basically. So I would just caution if you are doing that, just bear that in mind maybe and look for those differences and try to figure out a way to extract out those differences in your analysis first. Yeah.
Another question while you have a little bit of time. You know how when you look at the alignments, like in a genome browser, and you often see, looking across the gene, like if you've got a transcript assembled with no introns in it, you get like these peaks and valleys, right, in terms of the actual RNA read coverage. Is there any way to address that in the alignment, or and kind of what does it mean too? I've always wondered that, like if there are parts of the gene that are higher or lower than others.
You just mean it after aligning?
Yeah.
I think a lot of it comes down to, first of all, if there's repetitive regions, it might align somewhere else, and so it gets split across, even though it might have originated from that portion. The aligner doesn't know that, right? And so it might be aligning it elsewhere. It could also be that your reference is different from your reads a little bit. So unless you created your own reference out of the RNA-Seq reads, if you're using a different reference, there could just be SNP differences. And the aligner is going to go through and possibly not align as well if there's too many differences in that region. GC content comes into play as well. There's a number of reasons why you see such variability across transcripts. And yeah, I mean, there's normalization that takes place when you run any of these that tries to take that into account, but it's never 100% perfect.
Yeah, along those lines, so the only way you would know that specifically, right, would be if you were aligning to a genome, so that you knew like all these exons are like in this one gene, and some of them are higher and some of them are lower, if I interpreted what he was saying right. So if you don't have that information and you're just like annotating based on like Unipro or something, and you have all these contigs that are coming back as like the same gene, is there a way to like normalize expression like for a gene instead of having like 20 contigs that are all supposedly the same gene?
By
And they
contigs,
all
you mean-
have different values of expression, you know?
Right. And by contigs, you mean like different transcripts, isoforms that were assembled?
Yeah, they were aligned to like Trinity contigs, like.
So you're asking, is it possible to figure out which transcripts those reads came from?
No, I guess what I'm thinking of is if, I don't know. Maybe we can chat later,
Sure.
actually.
Yeah, I'm sorry, I'm just not
No,
understanding.
no worries. Thank you.
Todd's first question regarding before or after herbicide application. Contact herbicides will typically destroy the tissues pretty fast. So what are your thoughts in terms of adjusting the timing after herbicide application, how we could do that best in your opinion?
Well, so for contact, my suggestion would be if you really want to treat and get an idea of how resistant each plant is, I would suggest more of a cloning technique to be able to do that. Because even for a contact herbicide, I guess you're suggesting maybe pull from like another leaf or something instead. That could work. I would still expect to see some differences in the plant in terms of expression, just because it's a single system, right? And even if it doesn't look like it had any differences occurring, it could have expression differences occurring. Like I said, I really would suggest, if you can, to make clones and then spray those to get an idea of the level of resistance for each one of your plants rather than taking from sprayed tissue. Of course, there's drawbacks to that as well, because cloning itself might introduce some changes too, but it at least gets around the whole death process expression differences issue. Yeah.
So I heard this single cell technologies and the omics, so like genomics, transcriptomics, and metabolomics. So have you done anything like with the single cell technologies?
I personally have not,
Okay.
but I know that's like really developing, and there's
Yes, it's a pretty new technology.
yeah, there's so many more tools out there available for that now too.
Okay.
It's really becoming more and more commonplace.
Okay.
I personally don't have experience in it, so I can't speak much to it.
Okay.
But yeah, it's really exciting.
No, I just had that question very recently, so because I'm also new to that technology, so
Yeah.
I have no idea. So I thought.
No, I
might
wish I
be-
was.
yeah.
I think that's really
Thanks.
cool. Yeah. Okay, guys, if there's no more questions, I think we can end early and, yeah, leave for the day.